Good evening students, welcome to the circulatory system part 3. Previously we have discussed about the all components of the blood. Today we will discuss about the blood group system. Today we are going to discuss about blood group system. Blood group system. So as already we have discussed, we are having ABO type of blood group system. What kind of blood group system we are having? We are having ABO type of blood group system. According to this blood group system, we have the blood groups as A blood group, B blood group, AB blood group and O blood group, right? So the concept of blood grouping was first discovered by Carl Landsteiner. Who was that? Carl Landsteiner. And RBCs of human beings have specific proteins. So RBCs of the human beings are having what? A specific kind of proteins. That specific kinds of proteins we are calling as what? Antigen. What we are calling? They are calling as antigens. These antigens are present upon the red blood cell surface. Where they are present? They present on the RBC surface. Okay. Against to this, what antibodies also present? Those are also considered as a special protein, and these antibodies are present inside the plasma. Where it is present? Inside the plasma they are having. So what in human beings, there are two type of antigens are there. Even not only antigens, even antibodies also two types are there. How many types are there? Two types of antigens, and at the same time two types of antibodies also present. So what are that antigens? That is antigen A and antigen B. The same way antibodies also two types, that is anti A and antibody B. So like this, total two types of special proteins are present. As already we have discussed, antigens are present on where? On the RBC surface, and these antibodies are inside the plasma, means in the RBC inside the plasma these antibodies are present. So here we have just discussed, we are having the blood groups as A blood group, B blood group, AB blood group and O blood group. So this is according to the ABO blood group system. So we will see what is this A blood group is having. So this A blood group has what? Antigen A. What kind of protein it is having? It is having antigen A. Antigen A proteins are present upon the surface, RBC surface, and complementary to this, antibodies are present. Which kind of antibody it is? Antibody B. Which kind of antibody it is? Antibody B is present. Where it is present? Inside the plasma it is present. Coming to the blood group B. So in the blood group B, antigens. Which kind of antigens? Antigen B present upon the RBC surface, and complementary to this, what? Anti-A type of antibodies are inside the plasma they are having. Coming to the AB type of blood group. In the AB type of blood group, antigen A protein, antigen B protein. These two proteins are present upon the RBC surface, and here in the plasma there is no antibodies are present. Coming to the O blood group. So in the O blood group, upon the RBC surface we cannot identify any kind of antigen proteins, but inside the plasma we can identify antibody A and antibody B. Okay. So these are the main characteristic features of blood groups. So antigens are present on where? Upon the RBC surface. Antibodies are present on where? In the plasma of the RBC. And see, antigen A, agglutination means here the clumping. So as in the last topic we have discussed, for the blood transfusion we need what? Actually same kind of blood group we required, means matching type of blood group we required. So means here if we have transfused for example take blood group A, if we have transfused the blood group B, what will happen in them? Those who are taken, the recipient, those who are taken, the recipient within as or within fraction of seconds. We might have seen their blood, in their blood what we can identify, this coagulation or clumping of the RBC can be occurred. So, this is called as what? Incompatibility. What we are calling? Incompatibility or mismatching kind of blood grouping. Okay. So, what? Antigen A and antibody B. So, clumping is occurred or this coagulation is occurred between what? Antigen A and antibody A. So, between these two, what is happening? Clumping is occurring or agglutination is occurring. So, while we are transferring the blood, it is very, very important whether that blood group is compatible or not. So, matching also called as what? Compatible. Clear? See here, very clearly we can see. So, this antigen A means what? It is which kind of blood group? It is A blood group. It is what kind of blood group? It is A blood group. So, upon the body surface what are present? Antigen A present and antibody A are present in the plasma, means it is which kind of blood group, ma? This is B blood group. What is that? B blood group. Antibody A is present in where? In the blood group B. So, here there is no matching, there is no compatibility of the blood group. So, because of that, what is happened? Because of the incompatibility, transfusion of blood occurs what? Agglutination or what is that? Clumping, means destruction of the RBCs we can see. So, this condition may leads to the death also, right. So, ABO system of blood grouping. According to that, how many blood groups we have? A, B, AB and O, right. So, O group we can consider as the universal donors. What we can call? Universal donors. And coming to the AB blood group, we can consider them as a universal recipients. What we can call? A recipient, means these AB blood groups can take from all kind of blood groups. This O blood group can donate the blood, can give the blood to the all kind of blood groups. See here, O can give to the A blood group and also B blood group and also AB blood group. Clear? So, O is considered as the universal donor and B is considered as the universal recipient. They can take this O can be gives to whom? To O also it can give, to A also it can give, to B also it can give, to AB also it can give. Coming to the universal recipient, it can take the blood from whom? From the AB type of blood group, from A blood group, from B blood group and OB blood group. So, a person with A type can receive the blood from whom? From A and O only. So, blood group A can receive the blood from whom? From A blood group and O kind of blood group only. And coming to the B blood groups also receive the blood from B blood group and O type of blood group only. Right, here you can see very clearly, again very clearly in the tabular form we can see A is the donor. So, it can donate to whom? To the A blood group it can give. It cannot give to the B blood group and also it can give AB blood group and it cannot give to whom? O blood group. Coming to the B. So, it cannot give to A blood group. Why A blood group? Because here what are present, ma? Antibody B are present. Okay, antibody here, sorry, antigen A is present. What is present here? Antigen A. So, here antigen A. Here what is this? Antigen A and antibody A. So, here clear, right. So, B blood group can be given to whom? They cannot give to the A blood group, and here the B blood group can be given to B kind of blood group, and to the AB also can give, and they cannot give to the O blood group. Coming to the AB blood group, they cannot give to A blood group and they cannot give to B blood group. AB can give to the AB kind of same kind of blood group and they cannot give to the O blood group, and universal donors. O is a universal donor. So, as it is a universal donor, it can give to all kind of blood groups. I think you might have understand. And what is RH system of blood? The same way how these upon the RBC, how a special protein, that is antigens, are present, the same way RH type of antigens also present. What are present, ma? RH type of antigens are present on the RBC surface. It is this RH antigen and RH factor also having other name, that we are calling it is antigen D. What is the other name? Antigen D or D antigen. What we can call it is D antigen. So, those RBCs are having with this antigens, we call them as RH plus. What we can call them? RH plus. The RBC, those who do not have the RH plus means RH type of antigens, they are considered as what? Negative, that is RH negative. In them antigens are absent. Clear? One more time I am telling. RH positive means the person, means the specific kind of these proteins which are present upon the RBC, they are considered as what? RH plus, RH positive, and those blood groups or else those RBCs, the person's RBCs, those who do not have this antigens on the surface of the RBC, they can considered as RH negative. They can be written in the form of RH minus and RH plus, but instead of plus and minus we should call it as positive and a negative group. Here you can see when RH positive blood is transferred to a RH negative blood. So, here the blood is compatible? No, opposite. It is not at all compatible. Incompatibility, mismatching is taking place when RH blood is transferred to the RH negative person. What is happened? Because of to this antigens, this negative blood group person, blood will be get sensitized and start forming the anti-D, means that is antibodies, they form against to this RH positive blood group in the negative person. It causes the agglutination, again coagulation also here, sorry, this blood clumping. What is happening here? Blood clumping or agglutination we can see. What we can see? Agglutination or coagulation we can see, means while the blood is transforming, transfusing or giving to some other person. So, what has to be matched? The compatibility of the same kind of blood groups, and positive or negative, means positive has to be given to the positive only, negative has to be given to the negative blood group only. If it is not happen, again what occur? Clumping of the blood will be occur. Is this clear, students? Yes. So, that factor is affecting in whom? See here we can see the RH incompatibility in pregnant ladies. So, here what? Mother is RH plus. Mother is what? RH plus. Mother and father. So, here father is the RH negative when the baby is first in the first pregnancy. The baby is with which kind of blood group? That is with the RH negative. What is this? RH negative, first pregnancy, RH positive. So, here you all can see in the first, okay, in the first pregnancy what happens? Delivery can be taken place in normal, but while during the delivery time, so placenta will be get cut. So, because of that some of the blood can be entered into the mother's body through the placenta. So, because of this, so here opposite blood groups are there, one is positive and other one is negative. So, because of this condition the mother body will be get sensitized and it starts producing the anti-D antibodies against to the which blood group, ma? Negative blood group. So, nothing will happen in the first time pregnancy, but in the second pregnancy if the same condition occurred with the same RH negative. So, what will happen? So second time maybe the fetus can be get died. Why? What is the reason? Here already mother body gets sensitized at the time of delivery, right? So already in the mother body what? Anti-D antibodies are produced already there. So they will be transferred, these antibodies are transferred through the placenta and it leads to the destruction of RBCs. What happens here? It causes the destruction of the RBCs and also severe anemic condition we can see, jaundice also we can see, and also heart failure conditions also we can see, and hydrophytosis means it's very severe condition, it leads to the death and may leads to the fetus of newborn. So this condition we called it is what? HDFN. What is the meaning of HDFN? H for hemolytic. What is that? Hemolytic disease of fetus or newborn. We can call it as fetus or newborn. Okay. So what is the preventive measurement for this means? Already at the first time only the child will be gone through with the blood group testing. So at that time only it will be clearly it will be clearly mentioned that they both are having different blood groups, means it is very clear information that this mother body already got sensitized. So so when at the time of or else after the delivery the mother has to be what? It has to be given what? Anti-D immunoglobulins in the form of injection. The mother has to be given after the delivery what? Anti-D immunoglobulins. So it prevents the formation of more antibodies in the mother's body. So in this way in the second pregnancy child can be saved. So here you can see, yeah, important points what you have to remember in this topic means RH antigen, determination of what? It determines RH factor, RH plus antigens are present on the RBC surface, and generally these RH negative antigens are absent, means in them nothing will be there, but when the body is exposed to the opposite blood group, so it immediately starts producing the antibodies against to the RH plus. Okay. So here they have mentioned very clearly, right. So this is called as what? RH incompatibility in pregnancy can be dangerous to the fetus or newborn. So RHD injections given to the RH mother prevention from what? Sensitization and protect the future pregnancies. So this injection has to be given immediately after the delivery, otherwise what will happen? So because of this, because of the cutting of the placenta only, mother body will be get sensitized and it starts producing the antibodies. Okay. So we will see some questions, students. Granulocytes are what? Lymphocytes and monocytes, are lymphocytes and basophils. Already we have discussed that is what? These WBCs are how many types? Two types. One is agranulocytes and the second one is granulocytes. What are they? Granulocytes. So these granulocytes are again three types, agranulocytes are two types. So what are these granulocytes? Already we have discussed eosinophil and basophil. The third one is here, what is this? Lymphocytes, and these are monocytes. What are they? And this third one is neutrophils. Neutrophils. So according to this, agranulocytes are, this is the correct answer. What are they? Lymphocytes and monocytes. White blood cells engulf the bacteria, means they are eating the bacteria and killing inside the cell. So that process we called it is diapedesis, means what we have discussed in the previous classes. If the RBC comes out of the blood vessel, we called that process is diapedesis, right, crossing the blood vessel. And phagocytosis means killing, engulfing the bacteria. So this is the correct answer. And active transport means if any substances want to transport required energy, that we called as what? Active transport. Coming to the passive transport means without any energy, if the substances will be transport, that we called it is passive transport. So for this question, correct answer is the B option. I hope you all understand, students. Right, coming to the circulatory system in our body. So in the earlier class also we have discussed the circulatory systems are how many types? Open circulatory system and a closed type of circulatory system. In the open circulatory system, what will happen? The blood flows from the where? From the heart to veins and arterioles, and directly they entered into the body cavity. Here in the closed circulatory system, from heart to the arteries and arterioles, and coming to the venules and veins, and again to the heart. Means what? Here the blood is transporting only in the blood vessels only. So that's why it is called as what? Closed type of circulatory system. So here in human beings we'll see closed type of circulatory system. So the blood circulatory system is having what? Heart is present, arteries are present, veins are present, and along with that blood capillaries are present. These are arteries and venules only, furtherly divided into smaller tubular structure. We call them as the capillaries. Here, right, what is heart? See, it is located in where? Between the two lungs. Where it is present? Between the two lungs it is present, and it is slightly tilted to the left side. So that's why everybody feels that this heart is located in the left side, but it is not correct. Exactly, it is located between the lungs, but it is slightly tilted towards the left side. So this is the reason the lungs are having what? Left side are having only 2 lobes, and right side lung is having how many lobes? With the 3 lobes. Means some of the space is occupied. That is the reason. And the heart is right center between the 2 lungs above the diaphragm. Right, between the below the lungs, what is present? A dome shaped diaphragm is present. Above it, this heart is located. Right, already we have discussed the narrow end means this end is slightly tilted towards the left side, and triangle heart is present. During work or concentration of the heart is mostly powerful at this end, giving feeling that the heart is on the left side. So this kind of when we are working hard or during the contractions, so we feel that heart is present in the left side, but it is not at all exactly correct one. Right. So what is the length of this heart? Means it is present 12 centimeters in length, and width is how many? Only 9 centimeters width, and everybody's heart is with their clenched or closed fist. Okay, ante every cheti pidikilanta valla heart untadani din meaning. Okay, and heart is covered with what? Pericardium. What we are calling? Pericardium. The entire heart is covered with a layer, we call that is pericardium. Double layered pericardium. So it is covered with the double layer. So outside we called it is outer pericardium, inner we called it is inner pericardium. Between them what will be there? A fluid like structure is there, that we called it is pericardial fluid. So this fluid helps from the external frictions, or it can be act as the shock absorber. Okay. So the summary, what is the summary? The circulatory system is having heart, arteries, veins and capillaries. It is situated in the chest region, okay, and where between the lungs and above the diaphragm, and it is triangle in shape and pointed towards the left, and this is what length is 12 centimeters, width is 9 centimeters, and heart is covered with the double layer, we called it is pericardium. So here you can see a fluid is there, this is called as pericardial fluid. It prevents from the mechanical injuries. Right. So our heart is how many chambered heart, students? 4 chambered heart. How many chamber? 4 chambered heart. What are that 4 chambered? 2 are atria and 2 are ventricles. What are they? True are atria and ventricles. Atria also called as what? Auricles. We can call them as auricles. Here you can see. So, this is a right atria, left atria, a right ventricle, left ventricle. So, this is called as what? Superior vena cava, okay, or else precaval vein. We can call it is precaval vein, okay, and downside whatever the tube-like structure is there, we called it is postcaval vein, postcaval vein, or else inferior vena cava. What we can call it is inferior vena cava. Okay. So, we have already discussed upper two chambers are called atria. Okay. What are the atria? Singly we called it is atrium. Two are ventricles, atria also called as auricles. So, these are having what? So, this right atrium, completely right side we can identify deoxygenated blood. Which kind of blood we can identify? The right side of the heart chambers, that is right atrium and right ventricle. These two have deoxygenated blood, and complete left side we can see, that is what? Left atrium and left ventricle. So, these two are having oxygenated blood. What kind of blood, students? That is oxygenated blood we can identify, and this heart is made with the myocardial tissue. What it is? It is formed with the myocardium tissue, and comparatively these two atria, ventricles are longer, auricles are smaller in size, okay, and these auricles are thinner and these ventricles are thicker in wall. Okay. So, the blood vessels which are entering into the heart. So, which vessels entered into the heart means, that is, as we all discussed just now, anterior vena cava, posterior vena cava. Anterior means this is superior vena cava and this is inferior vena cava. So, this superior vena cava bringing the deoxygenated blood. Which kind of blood it is bringing? Deoxygenated blood is bringing from the upper side of the body parts. Like which parts? That is from the head region, neck region, chest and arms region. From these all parts, deoxy blood will be collected, and through the inferior vena cava, superior vena cava or else anterior vena cava, it entered into where? It entered into the, what it is? It is the right atria. Okay. And see, from the down parts also brings the downer half of the body, means which parts? That is abdomen, pelvic region, and from the legs region deoxygenated blood collects, and it can be transported through the posterior vena cava, and it enters into where? Again into the atria itself only. Clear, students? Okay. So, that anterior and posterior, they are bringing the blood into where? Into the heart region. And what are these pulmonary veins? So, two veins are from each lungs. Here you can see, this is what? This is right side and this is left side. Two left and two right. Total 4 pulmonary veins are there. So, these 4 pulmonary veins are bringing which kind of blood? They are bringing oxygenated blood. So, this oxygenated blood transports which side? Away from the heart, right? They have to be transported to the different kind of body parts. Okay. Here you can see very clearly, arising from which one? That is pulmonary artery. Here you can see, this is the pulmonary artery. So, this pulmonary artery is taking which kind of blood? Deoxygenated blood. From where? From the ventricles. From the ventricles through pulmonary artery, the deoxy blood sending into where? Sending into the lungs. For what purpose? For the purification purpose. Okay. Next vessel, that is what? Aorta. Here you can see very clearly, this is aorta. So, this is aorta. This aorta is originated from where? From these left ventricle. From where? From the left ventricle it is originated. Here which kind of blood is there? Oxygenated blood is there. Okay, so here oxygenated blood, and this through the pulmonary vein oxygenated blood enters into the which? Left atrium. From the left atrium to the left ventricle it goes, and from the left ventricle through the aorta it goes to the different kind of body parts. Okay. Next, coronary arteries. So, where these coronary arteries are present? See here, it's very clearly mentioned over here in the diagram. It is very clearly mentioned. These coronary arteries are arising from the base of the aorta, right? So, these two are the atria and these two are the ventricles. So, just below the auricles or atria, what are there? Coronary arteries are there. So, these coronary arteries are bringing or supplying the blood to the heart muscles. What they are doing, ma? They are supplying the blood to the heart muscles. So, if any blockage, here you can see a plaque occurred. So, this plaque may be because of the deposition. What is that? Deposition of calcium, or maybe deposition of fats. So, what happened? So, this or accumulated plaque is accumulated and the passage become narrow, means the blood supply is become less, means not proper supply of the blood is not there. So, it causes what? Deadening condition. What it is causing? Deadening condition, no proper supply to the heart muscles. So, because of that what happened? Myocardial infarction or else a heart attack can be occurred. Generally we call it is heart attack, and myocardial infarction is a scientific term. So, it is because of the plaque or else any fat deposits occurred in these coronary arteries, and these coronary arteries are raised just below from the base of the arteries. So, what is the main function of this coronary arteries? Pumping the blood to the heart vessels. So, because of this blockage we can experience what? Heart attack. Okay. And cardiac veins are, they are draining what? Deoxygenated blood from the heart muscles. Coronary arteries is pumping what? Oxygenated blood, and coronary veins are deoxygenated blood, they are pumping deoxygenated blood from the heart muscles. Okay. So, what is angina pectoris? So, angina pectoris is what? The heart muscles collect blood from the heart walls and pour into the right auricles. Into the right auricles, the heart muscles are collecting the blood from the heart walls, and where they are pouring? They are pouring into the right auricle. Angina pectoris is the chest pain due to insufficient blood supply to the heart muscles. So, here we can see two kinds of disorders we can identify. One is angina pectoris and the second one is myocardial infarction. So, these two heart diseases are caused by the improper supply of the blood to the heart. Clear? So, heart can be considered as a bank cashier. You know, I think you might have heard this word, bank cashiers. They can take, means in their hand lot of amount of money they can seal, but all the money they can be used for their own? No, they cannot. Only salary they have to use. Their salary, whatever the salary they are getting, that they have to use. Maybe that salary sometime it is not sufficient. So, at that time they may face some problems. Just example purpose it has given, that's it. No relation to the bank and heart. Clear, students? Right. So, what are the valves are regulating the blood flow? So, as we know that valves ka what is main function? It functions, or the main function is only single flow. Okay. They prevent backflow. What they can do? Backflow they can prevent, or else they see that single way of flow, single way of blood has to be transported. So, this is the function of the valves. So, how many valves are there? We will see. Okay. So, these are two what? Arteries, right, and these two are the ventricles. So, in this atrial region and here, and between the atria and the ventricle. So this is which side we have discussed? Which side it is? This is a right side and this is left side, right? So in the between the right atria and the ventricle, there is a wall that is called as a tricuspid wall. What we are calling it is? It is calling as tricuspid wall. Okay, and here in the left side ventricle. So here in the left side, what we can see? Between the atria, left atria and ventricular, we can identify bicuspid wall. What we can call? Bicuspid wall or mitral valve is present. What is that? Mitral valve is present. So the function of the valve is mainly allow the single direction of the blood flow or else stop the back flow of the blood. So while the blood is transporting from atria to the ventricles, so it allow the passage, right, from these ventricles. If the pressure is occurred, more pressure is there in the ventricles, it stops the back flow of the blood from ventricles to the auricles. The same thing, tricuspid and bicuspid both do the same kind of function. Okay, see here, so this is tricuspid wall, right? Cusp-like structures are there, and these cusp-like structures are maintained or can be held by a thread-like structures. They are called as chordae tendineae, and these thread-like structures are originated from the papillary muscles. From where these papillary muscles are originated means, here you can see these ventricular walls muscles only projected into a finger-like structures. These finger-like structures only called as the papillary muscles. Upon these papillary muscles, thread-like structures are there. They are called as chordae tendineae. They are holding these tricuspid and bicuspid walls. Both they are keeping in their proper position because of this chordae tendineae. Okay. So next, what is the main function of the bicuspid wall? So here we have already discussed the blood flow of the from left atrium to the left ventricle. So single flow and also prevent the back flow. And here what are there? Tricuspid valve is there. It is also allowing the blood flow of from the what is this right atria to the right ventricle. So the same way here also tricuspid and bicuspid valves are protected with the chordae tendineae. Along with the tricuspid and bicuspid valve, other valves also there. Those are called as pulmonary semilunar valves. What is that? Actually semilunar valves. What are they? Semilunar valves. These semilunar valves are two types. One is a pulmonary semilunar valve and the second one is aortic semilunar valve. Pulmonary semilunar valve is present where this deoxy blood is taken into the pulmonary artery, means in this region it is present. So when these ventricles get contract, the blood has to be flow into this pulmonary artery. Okay, and here in this which region? In the left ventricle. So from the deoxy blood it is going to where? Into the aorta, and in this region which is present, aortic semilunar valve is present. When these ventricle is get contracted, it will be get open, right? And when it close means when the atria is pumping the blood to the ventricle, it will be get closed. So these are the different kind of valves we can see. Coming to this atrial systole and a ventricular systole. Systole means they are get contracting. What is happening? Contraction is happening. In the diastole, we can identify the chambers are getting relaxed. What is happening in the diastole? They got relaxed. So here what is happening in the atrial systole? As I have mentioned over there, these atrial muscles are getting, atrial systole means what I said? Contraction. They are getting contraction. Because of this contraction, what is happen? Vena cava and pulmonary veins get closed and blood enters from ventricle crossing to the tricuspid wall and bicuspid valve. Means simultaneously these two auricles at a time get contract. Because of that the blood flow is happening from the auricles to the ventricles. So at this time what has to be open, students? This tricuspid valve and bicuspid valves has to be get open. So because of this what is happen? So at this time what has to be these are open, but what has to be get closed? Semilunar valves has to be get closed. So when these open, semilunar valves will be closed. So because of that closure of this what pulmonary artery, semilunar valve and aorta semilunar valve, dub sound is produced. What is produced? Dub sound is produced. And in the ventricle systole, what is happening? These ventricles are getting contraction. When these ventricles are contracted, what has to be closed? Here the tricuspid and bicuspid has to be closed. So because of that lub sound is produced. And what has to be open? Here the semilunar valves has to be open for the flow of the blood. Okay, right. So because of this contraction, the blood is atrial contraction, the blood is transporting to the ventricles. When these ventricles get contract, these two chambers will be relaxed and here tri and bicuspid valves to be gets closed, and because of this contraction these semilunar valves to be open. These points are very very important. What is closing, what is opening for this blood pumping you need to remember. Okay, atrial contractions lo emaitundi, ventricles అనేవి రిలాక్స్ అవుతున్నాయి. వెంట్రికల్ కాంట్రాక్షన్స్ లో ఏమవుతున్నాయి? అట్రియాస్ అనేవి రిలాక్స్ అవుతున్నాయి. ఎందుకంటే 4 చాంబర్స్ ఒకటేసారి కాంట్రాక్షన్ కానీ రిలాక్సేషన్ కానీ జరగదు. రెండు అట్రియాస్ కాంట్రాక్ట్ అవుతున్నప్పుడు వెంట్రికల్స్ రిలాక్స్ అయితే, వెంట్రికల్స్ కాంట్రాక్ట్ అయినప్పుడు పైన ఉన్నటువంటి రెండు అట్రియాస్ ఏమవ్వాలి? రిలాక్స్డ్ పొజిషన్లోకి రావాలి. క్లియర్ స్టూడెంట్స్? సో for these blood flow, semilunar valves and tri and bicuspid valves play an important role. See, as we have already discussed, inside the heart valves are there. That is which kind of valves? Tricuspid valve and bicuspid valve and semilunar valves. What are the semilunar valves we discussed? Pulmonary semilunar valve and aortic semilunar valve. All these are, see, they are supported with what? They are in the proper positions because of this cardiac tendinae only. So they are present upon the papillary muscles. This already we have discussed very clearly, right? So heartbeat and heart sound. So with what is the time to complete one cardiac cycle? Means that is 0.8 seconds only. In how much time it is completing one cardiac cycle? Means that is 0.8 seconds. What is cardiac cycle? The first heartbeat of the heart and the next heartbeat, it is called as the cardiac cycle. So what kind of sequences can be, events can be taken place in a sequential order? Means that is atrial systole, ventricular systole, joint diastole. So these three events are occurred in single cardiac cycle. So it can be completed in 0.8 second. Atrial systole is taken only what? 0.1 second. In the atrial systole, what are contracting? Atrias are contracting, right? After that, what has to be get contract? Ventricles has to be get contract. It can be taken place in how many minutes? That in how many seconds? That is 0.3 seconds. So 4 chambered at a time for a while. Okay, that is what? 0.4 seconds. All diastole means all chambers will be get relaxed. That is called as what? That is called as the joint diastole. Clear? So remaining we will see next class.